Fasting strengthens the immune system to fight cancer. Fasting can help your immune system fight cancer. New research conducted on mice has shown that fasting effectively reprograms the metabolism of specific immune system cells, which makes them fight cancer more effectively. However, the study authors do not recommend fasting without consulting a doctor, especially for sick people. Fasting and other dietary regimens are increasingly being studied as ways to deprive cancer cells of the nutrients they need to grow. The impact of fasting and diet in general on increasing the effectiveness of cancer treatment is also analyzed. In recent research, scientists from Memorial Sloan Kettering Cancer Center, MSK, have shown that properly planned fasting can reprogram the metabolism of the so-called NK cells, helping them survive in the difficult environment within cancer lesions, as well as improving their ability to fight the disease. The findings may help explain one of the mechanisms by which fasting may help the body defend against cancer. Fasting can reduce fat and improve metabolism, which can also be significant. And although more research is needed, the results suggest that fasting may be a strategy to improve the immune response in the fight against cancer. The study appeared in Immunity. What are NK cells? NK cells, natural killer cells, are a type of white blood cell that can eliminate abnormal or damaged cells, such as cancer cells or virus-infected cells. They get their name because they can destroy a threat they have not previously faced, unlike T cells, which require prior knowledge of the enemy to trigger a targeted response. Generally, the more NK cells are present in and around the tumor, the better the patient's prognosis. The tumor is very hungry, says immunologist Joseph Sun, co-author of the study. It absorbs essential nutrients, creating a hostile environment, often rich in lipids that are harmful to most immune cells. In our research, we show that fasting reprograms NK cells to better survive in this environment, he adds. In their research, scientists focused on rodents. Mice previously injected with cancer cells were placed on a diet, depriving them of food for 24 hours twice a week. Between fasts, the rodents were allowed to eat freely. As expected, the mice did not experience overall weight loss, but there was a decrease in blood glucose levels, which triggered an increase in free fatty acids, which are lipids released by fat cells that can serve as an alternative source of energy when other nutrients are lacking. During each of these fasting cycles, NK cells learn to use these fatty acids as an alternative fuel source. This optimizes their anti-tumor response because the tumor microenvironment contains a high concentration of lipids, Sun admits, adding that after cycles of fasting, NK cells were able to enter the tumor and survive thanks to this metabolic training. The researchers also observed that fasting led to a redistribution of NK cells in the body. Many NK cells migrated to the bone marrow, where, due to food restriction, they were exposed to high levels of a key signaling protein called interleukin-12. This primed NK cells to produce more interferon gamma, a cytokine that plays an important role in anti-cancer responses. Meanwhile, NK cells in the spleen were undergoing separate reprogramming, making them better able to use lipids as a fuel source instead of glucose. By combining these two mechanisms, we found that NK cells are pre-primed to produce more cytokines within the tumor, says Rebecca Delconte, co-author of the study. And thanks to metabolic reprogramming, they are able to survive in the tumor environment and have improved anti-cancer properties, he adds. It is not yet clear whether there are two separate populations of NK cells that are trained differently in different parts of the body, or whether the cells eventually pass through both places during their several-week life cycle. That's the million-dollar question. And one of them we have only just begun to answer with the cell labeling techniques that we used in this study, says Dr. Sunday. Scientists say there are several potential opportunities to expand research on the mouse model into the clinic. First, clinical trials are already beginning to test the safety and effectiveness of fasting in combination with standard existing cancer treatments. Another possibility would be to identify drugs that could act on the underlying mechanisms established without requiring patients to fast. Third, metabolic training could take place outside the body, and then such trained NK cells could be introduced into the body to improve treatment effects. Currently, however, more data is still needed on the effects of fasting on people with cancer. There are many different types of fasting, some can be helpful and others harmful. Patients should talk to their doctors about what is safe and healthy for their individual situation, says Neil Iyengar, an oncologist at MSK who was not directly involved in this study.